way.
Great. Thank you so much. And thank you, Harry, and the whole committee for the invitation to speak for the Evolutionary Anthropology webinar series, University of Liverpool. I was just joking with them that I was hoping to actually be there, but unfortunately, COVID had other plans for that. So today I'm going to talk about some of the history of the discovery of Homo naledi, what it looked like, what that species looked like, and then some of the newest finds and some of the implications that we have for the finds for Homo naledi. So the Rising Star Cave system is where Homo naledi was recovered from. I have that on the star there. And this is an area of South Africa known as the cradle of humankind. And so you might be more familiar or somewhat familiar with some of the other more famous fossils from there, or famous fossils. This is the Tong child from the site of Tong. That's Mrs. Plez, or Australopithecus africanus, one of my all-time favorite fossils. This is Australopithecus robustus. It's found at Sparkons and Cooper's Cave. And then Australopithecus sediba that we recovered in 2010. And so these are all great famous fossil sites. And again, the Rising Star Cave system is down here on the lower left. So there were some recreational cavers that were exploring the cave system, as they want to do in South Africa, especially in this region, and they came across some fossils. They then got a hold of Lee Berger at the University of Witwatersrand and told them, hey, they look interesting. So Lee sent some of his cavers down from the university to look at them and realized that this was a big deal, that there were many different fossils that are found in this one chamber that they came across. But it's in these really hard to reach caverns and fissures. And so Lee put out a call literally on Facebook to say that he needed small bodied hominins to go down and excavate the remains. He got probably about 100 applications. This was in 2013, and he ended up settling on 5 women and 1 male, and the male dropped out. So it ended up being 6 females who the media dubbed the underground astronauts. But you can see, here's one of my colleagues from the University of Witwatersrand, so how he's sliding through these really small crevices in order to get to the fossils. This is a picture of the top of the cave. So if you were looking at the gradient of the cave and then you're looking at tips, so there's not really anything to see, but I figured throw a picture in there anyway. So they started excavating in 2013, and then they excavated for a couple weeks again in 2014. So this is a caricature, kind of a cartoon picture of what the original excavation, the original chamber looked like. And this is from the Dirks paper that talks about the taphonomy and the geological context. So you can see the fossil concentration is quite dense. And so the first pass, they went and excavated the surface of the chamber. But then they found that the fossil concentration was so dense, they had to even switch to barefoot excavations. And then there was a small area in the middle that had some long bones sticking out. And so from there, again, this is the caricature. But from there, they decided they were going to concentrate in this one section in order to make sure that those bones got preserved well enough. And so the excavation shifted a little bit to just concentrating on this. It's kind of like a large coffee table sized area where they started to excavate. So again, there was about 2 weeks in the fall of 2013, and then another 2 weeks in the fall of 2014. And so here you can see that fossil concentration. In some areas, there's more bones than the sediment. And we can find some great association with some of the bones. So this is a foot on the bottom here. And so as you can see, here's the ankle with the calcaneus, the talus, here's your lower leg bones, the tibia and the fibula. Again, all right next to each other, so completely associated. So very, very strong context for those and association. After those roughly 4 weeks of excavation, they brought up just shy of 1,500 fossils. That's a lot of fossils for a very short period of time. And so while they shifted excavations to that smaller section, there were so many fossils, it was basically enough. So let's see what we have here. Let's start to analyze that. And so that was Lee Berger's next step, was to decide, okay, we need to start analyzing these fossils and see what we have, because as they were coming out of the chamber, out of the cave, they're looking at them and parts of them look like a certain species, they have some similarities, but other parts didn't. Again, they needed a full-blown analysis. And this is one of the reasons I put in that, or I was thinking in that abstract about how the fossils were analyzed slightly differently than a lot of other fossils in paleoanthropological history. So Lee put out a call for early career scientists. They had either recently excavated or had large data sets, recently graduated or were still a PhD students that could have the data set to help to analyze these fossils. So you had these early career scientists that were analyzing them versus, again, people that had analyzed tons of fossils and other species, other primary analysts of other species. So that was very, very different from other ways of analyzing these fossils. He put out a call for these early career scientists. He got just over, I think, 300 applicants. He ended up settling, he's got 30 individuals from 16 different countries. And I was lucky enough to be a part of that because I had worked in South Africa. I did my master's as well as my PhD research down there. But then in 2010, I started working with Australopithecus sediba. And specifically, I work on the teeth, looking at the shape, the form of the occlusal surface and how that has changed over time, what it can tell us about evolutionary relationships. So since I had done a lot of primary research with sediba in 2010, I had this large data set to be able to help to work on the dentition for Homo naledi. So that's how I got involved in this. And so here's a picture of the individuals. This is not everybody, but this is the group that, a large portion of the group that did get to work on this. Lee put together a Rising Star workshop that was in that June of that year, June of 2014. And again, we had this great crew. So we had these early career scientists, but then some of these older individuals, more, let's say, experienced, that were also around to kind of guide, help, as well as contribute, obviously. So again, very different way of analyzing some of these fossils. So from this, we were divided up into our different groups and different areas of the lab. This is the lab at the University of Vaterjand. And so I was in an area known as the tooth booth. There was Handland, there was Hip Heaven. There was a whole area where all the cranial people were sitting. And so we would all sit in our small little areas and work all day on the fossils. And then at night, we'd all sit around at the pub and we would have our computers out and our PDFs, and we would talk about the things that we had found that day and research loads of extra things. So after roughly 6 weeks of analyzing the fossils that had come out of the cave, we decided that it was a new species. Again, each of the fossils looked like had similarities with other species. And that was the most interesting / frustrating part of it. Again, as we'd sit around and talk about it at night, one person would say, oh, it looks very much like Australopithecus, and others say, oh, no, there's no way, it looks very much like Homo. And maybe even Homo erectus, because it shares a lot of similarities with Homo erectus. Can we really justify calling it a new species? But with the suite of characteristics that came out of the species, there was no way that we could, it's not perfectly consistent with any other species. And therefore we settled on, we needed to name it a new species. So here's a picture. So I'm just going to go through some of the rough characteristics of what Homo naledi looked like. So we start off with the cranium. So we've got, this is Dinaledi hominid 1. As you can see, it's got a nice little brow ridge here, a little bit of forehead, but more of a globular shape. So it has a very modern or derived shape to the cranium, to the skull. And so that was very interesting. I really like this picture because it puts this National Geographic picture where it's got the imprint of what a modern human skull looks like. And so it's got a very modern, like I said, it's trending towards that globular shape versus that long, low vault that we see in, say, Homo erectus, the Australopithecines, and likewise. But so again, it's coming up more like a forehead. However, it has a very small cranial capacity, between 465 and 560. For comparison, we have modern humans average size of about 1,400. And so that's what we'll see with, say, the Neanderthals. So with these cranial capacity, it's quite small to have such a modern or derived globular shape to the cranium. And so this is some work that was done by Heather Garvin. She was a woman that she did her PhD looking at endocranial volumes. And so she was able to mirror image the cranium, that piece that we have. And then she was able to get this cranial capacity out there. So again, quite fascinating that it's that low of a number. So here's some more pictures of those. And again, this is part of Dinaledi hominid 1, is dentition. I always argue that this is the coolest part of the site, the dentition. We have 190 whole or fragmentary teeth, and they range in age. We have the deciduous teeth on your left. We have a large number of deciduous. That's probably my favorite fossil from the Dinaledi Chamber, at least. That's 1400. So this child, this is the lower mandible of a child. And here we have our Dinaledi hominid 1. This part is in mirror image, but we have the complete dentition of the mandible, part of the top of the maxilla. And then we have some really old individuals, much older individuals, where you can see their teeth are quite worn down from there. So just a little bit more, because this is really my forte. This is what I specialize in. So some of the characteristics about it. So we have a small anterior teeth. So the front incisors and canines, the smaller anterior teeth than Australopithecus. So they're very much like the genus Homo. We also have the post-canines, so your premolars or molars are more like early Homo. And a couple unique characteristics that we have are the canine has got a cuspule or a little cusplet, depending on the literature. And so this is this little mitten shape that we have on the canines. And we only see that in a couple other fossils. In Demnisi, it's on a couple of those mandibles, but not all of the mandibles. So that's a somewhat unique, again, with the suite of traits that we have for Homo naledi. And we've got the P4, it's got this extended talonid, it's got an extended back part of this, the fourth premolar. And also what is quite interesting is the molar size gradient. So for modern humans, our third molars are the smallest. The first molars are typically the biggest. 1 and 2 can be similar in size, but they typically decrease as you go back in the within the jaw within the mouth. But for the primitive condition is where the third molars are the biggest and the first molars are the smallest. And that's what we see with Homo naledi. So we have these quite modern, quite derived traits, but a primitive or ancestral-like molar size gradient. Again, a mix of interesting traits. So here's a picture of the tooth booth. I wanted to include in there for a couple different reasons. Sans the baby, obviously, but that's Matt Skinner. He's at the University of Kent right now, I believe. And we also had not pictured here Joel Irish Schwarzenegger. He's at Liverpool John Moores, so I thought I would make a shout out to him. But the other thing that we did in here that I think this picture shows well is that when we took each of the teeth, we tried to associate them. And some of them are found quite close to each other. Again, some of them not quite as close to each other. So we're looking at the depositional context to try to put them next to each other. And that's how that's what we're doing here. We're looking at things like the side, the size, the age of the fossils, what type of wear they have, and then the interproximal facets match up as they're next to each other. As one tooth is occluding next to another, it's going to leave a mark on that tooth, so you can look at what those interproximal facets look like. And ultimately, based on the dentition, we found that there were 15 individuals, a minimum of 15 individuals that were found, again, in that Dinaledi chamber. So again, some of these, this is 1400, that fossil I was just telling you about, and we can look at fossils on the other side. Again, the same age, roughly the same size, and then we've got these two tooth germs, these first molars that fit perfectly with certainly with 1400. So we estimate that this is all one individual, and all this is going to play very well into the taphonomy or the context and how they actually got into the cave that we'll get to here in just a second. So the last thing I want to say about the teeth is that a couple things. So one of the things that I look at is using elliptical Fourier analysis to look at the shape of the tooth. So just looking at the morphology, how does it compare to others? And so when I plot the mandibular P3s of Homo naledi, this is what it looks like. They're very consistent Very simple, very consistent. They look very similar to each other. And so this is not what we see with other species. So we say it has a very low intraspecific variation versus species or like Australopithecus africanus have a very large range of variation and also over a long period of time. So this one, we have a relatively short period of time that we know of so far. And it's all from, you know, this one locality so far. And again, they plot right on top of each other. So this suggests possibly some this related community, that they are related to each other because your teeth look more similar to the individuals you are related to versus someone that is that you're not related to, much further away. So I also looked at a relative cusp analysis. So this is looking at the different cusps of a maxillary tooth. So typically your maxillary teeth have 4 cusps. Typically the mandibular teeth have 5. And so, but the shapes and the sizes of these cusps change over time. And so that's something else that I recently put out in some of the some recent literature, just looking at the overall evolutionary relationship. And again, does not consistently plot with any current species that we have. So just a rough rundown of the rest of the body. So we've got to the shoulders. So we have part of the scapula or the shoulder blade. It looks like the shoulders were a little bit higher, a little bit more angled up. So this is the scapula, part of the glenoid socket, is more angled with Homo naledi than it is in modern humans. We've got the thorax, and so this area, middle area, is going to be more narrow towards the top of the cave. So that's going to suggest that it has more mobility in the shoulder girdle. That's something that is more similar to our non-human primate relatives than to modern humans. So we don't have as much mobility in our shoulder girdle as Homo naledi would have. So the arms, the muscle attachments of the lower arm going to suggest that it had the muscle attachment, that it was doing some climbing, that it was in the trees at some point. And we see that reflected in the hand. We have loads of hand fossils. Again, a complete hand. And so when we're looking at these pieces, it's got a human-like finger to thumb ratio, but it also has some curved phalanges. And so that's another thing that suggests that Homo naledi would have spent some time in the trees. Although it has some other modern characteristics, and some of the wrist bones are also quite modern, very suggestive of, again, more like us, more derived. The pelvis, to me, is another big deal about Homo naledi. So we have, the pelvis is very flared. So a lot of you are familiar, or maybe you've heard of Lucy or Australopithecus afarensis that's found in East Africa, dates to about 3 million years. And I put a modern human pelvis here underneath it. The pelvis of Homo naledi is very flared versus, again, ours is going to be more bowl-shaped. So morphologically, before we had a date for this, to me, that pushed back the date of these specimens to, we thought, roughly 2 million years old. Morphologically, it looked to be roughly 2 million years old. And again, when we first came out, there were several hypotheses that it was going to be quite old, based on the amount of time we think it spent in the trees, as well as that very, very primitive pelvis. So here is the femur. Got some angulation to that. So here's a picture, another picture, what those look like. So this is the chimpanzee femur on your right, in the middle picture. And then this is a modern human femur. So you can see how it's angled, and that's all related to our bipedalism. So you're gonna have a large femoral neck angle, and then the whole shaft of the bone is going to angle down, and so that your center of gravity is going to be underneath your, in between your knees. So this is not something that we see with a quadruped. So again, it's very, very indicative of being a biped, being a great, very efficient biped, and it's not unique. It's not one bone that we found, this one femur that looks like that they were a biped. We have multiple repeated elements that tell us that it was a biped and a consistent obligate biped, despite the fact that it has a pelvis. Again, that looks more primitive, not like a quadruped, but an individual that is transitioning towards full bipedalism. And so here's another picture of what that angle would be, again, underneath the body for the knee. That's your lower femur. So here we have some tibia, long, lean, that's your lower leg bones. Tibia, and again, the muscle attachments don't suggest anything except obligate bipedalism. And then we have the foot. We have a good colleague of mine, friend of mine, that was on the Rising Star Committee, works at the University of Witwatersrand. He used to be a podiatrist, transformed into a paleoanthropologist. And he said that if this foot walked into his office, he wouldn't bat an eyelid. It's very, very modern foot. It's got a little bit of curvature to the phalanges here, you can see, but it also has a little bit of curvature to the foot, and it's a little bit small. But otherwise, we're going to have it looks like a very, very modern foot. And we have loads of foot bones as well. So that's something that's also really exciting and not very common. And as I showed you in the picture earlier, they are somewhat, we have some that are associated directly with each other. And so it's very nice for putting together individuals. So I really like this picture just to emphasize the different types of characteristics that we have. Again, these Homo-like features that make the specimen very derived, but then some Australopithecine-like features that are more primitive, more ancestral-like. So again, the primitive shoulders, that flared pelvis to me is quite interesting, and then the curved fingers. But we've got the shape of the skull is very modern, despite the fact that it has a very small cranium. The hands, again, we've got some wrist-like movement that are very modern-like, but we'll also talk about here in just a second. Long limbs, everything suggesting obligate bipedal gait, and then the human-like feet. All right, so what does that mean about the taphonomy? So the taphonomy, anything that happened to the bone since the organism died, was it chewed on? Was it brought in? How was it actually brought into the cave? So looking at some of the site formation context. And so we look at the bones themselves, they have some snail and beetle modification. We've got 2 antemortem or pre-death fractures that are on there. Nothing that would suggest that an animal that the hominid died that way. And we have 1 specimen that has very mild normal arthritis. So that would be that part of what we think is part of that older individual, the teeth I was showing you, how they were worn down. So how did they get in the cave? We have no evidence of any diseases or disorders on the bones. That does not mean they didn't die of a disease, but not every disease is going to manifest itself onto the bones. But we have no evidence of that in any of the individuals thus far in the whole Rising Star cave system, not just at Dinaledi. We have no evidence of any carnivore or scavenger modification. So a lot of other cave sites we find in South Africa are going to have where carnivores, particularly leopards, are going to drag them into caves or drag them up into trees, and then they'll drop them, but they'll chew on them. We're going to see change in what remains that we'll have of the bone, as well as some carnivore tooth marks, some punctuate depressions that we'll find on the bones. And we don't have any of that in Dinaledi. The scavenger modifications, we'll see porcupines will chew on bones. They'll leave very diagnostic marks. None of that either. Excuse me. We don't have any evidence of water transport. And so this is something else that we see with other fossil sites in that cradle in particular, is that you'll see the heavy rains, washing, all kinds of materials in. So we'll get some bovids, you'll have some carnivores, you'll have some hominids that'll wash into the cave, as well as other debris, seeds, things like that. We have, like at Malapa, where Australopithecus sediba was found, it's a vertical death trap. So we have all kinds of things that have dropped in and fossilized into that cave area. But we have no evidence of this water transport. And one of the biggest ways to support that, one of the biggest lines of evidence to support that, is the fact that the collection is made up of almost entirely of hominids. So no scavenger or carnivore is preferentially chooses just hominids and is only going to place hominids in the back of this cave. We also, no water transport is going to preferentially wash in just hominids, right? A lack of this other debris. So to me, that is strong evidence for the fact that, you know, we don't have any water transport of the hominids. So the current hypothesis that we have, and I do emphasize hypothesis, but we have failed to reject it for 6 years now, is that the hominids were brought in, that they were deliberately deposited into the back of the cave. And some of the newest evidence provides even stronger support for this. but that an organism died and that they dragged it, they brought it back into the Dinaledi Chamber. And so this is what that would look like. Again, here's a little more of a caricature, but you've got this pathway getting back into the chamber. So it's about the length of a football field from the opening here to the Dinaledi Chamber. And again, this is much more complex. I'll show you another picture. This is simplified, which is good because it is very complex. But coming down this direction, they would have had to go through an area known as Superman's Crawl, which is quite small. They say it's about less than 10 inches in height. Now that area might not always geologically have been that small. We do think it was restricted in that section, but maybe not quite as restricted, but very, very small. So this is one of the spots where the cavers, they've struggled a little bit. It's crawling through that section. So they literally had to turn their head and then fly through like Superman would. And at the museum at Maripang in South Africa, they have a foam cutout. So you could try to squeeze through, see if you would fit through the little section. So once you get through that area, then you have to go up what's called Dragon's Back. So all these rocks that are jutting out and then down into the Dinaledi Chamber. From here, there were several hypotheses, but we think that it probably dropped the bodies here. That they probably weren't going in and placing the bodies, but it is slightly at a gradient. So maybe that's how they would have rolled down a little bit into this chamber to get into the whole section. But we do have bones that are found here, more concentrated, and then as they are in separate sections for that. So this is a lot to do to bring your dead to very far areas of the back of the cave. We don't have any evidence of any fire yet, but that's one of the questions. How did they begs the question, how did you get back there? How did you find that it existed? How did you know that? How did you find your way back there? And so we are looking for any type of carbon residue, for any signs of fire that we can find. But as of yet, there is no signs of this just yet. And I just want to point out one other thing. So think about the water transport. Water would have had to, again, preferentially move hominids down into this more restricted area and then fill the entire, this area, chamber here, up with water over Dragon's Back and then get it into the Dinaledi Chamber, another restricted section. And again, we don't have any evidence of that. It'd be much harder also to get water to transport chunks, partially articulated bodies to where they sit fossilized with, where they're associated with each other. It wouldn't be more commingled than they are thus far. So again, that's the current hypothesis going forward, how they got in there. So this is that is site 101. That was the Dinaledi Chamber, was site 101. In 2017, we found Lesedi Chamber, or we published on the Lesedi Chamber, and this is site 102. So again, this is the simplified version of what the Dinaledi Chamber looked like. This is the complex cave 3D version of what that looks like. So this is Lesedi is 102. So here's 101 over here, and 102 is in red back this direction. So it's a little bit more complicated, but it's this more open chamber where we have an adult male skeleton that seems laid out. We also have evidence for a child, so a deciduous individual who's got a deciduous tooth, as well as a deciduous tooth and part of a female skeleton. Also in 2017 is when we came up with the date. So we're published on the date. When we were originally in the Rising Star workshop, we did not have a date to work with. And that was both frustrating and exciting. So as you know, Lee was emphasizing to us how it can often bias. And as frustrating as it is, it can bias you, because you would think that it has to be, well, it has to be Homo if it dates to 300,000. It has to be Australopithecus if it dates to 2 million years, if it dates to 3 million years, right? You start to divide up where you think that fossil should go based on the date. And he very much, and the workshop was very focused on what does the morphology say to us. And so again, in hindsight, I thought it was great. At the moment, it was a little bit frustrating because it might have helped us. But the fact that that Homo naledi has such an interesting suite of characteristics already, you weren't able to pigeonhole it into a very specific region, a very specific time frame. But 300,000, roughly 300,000, is completely unexpected. We did not expect that. And there were 10, we did 5 different dating techniques, but from 2 different labs each. So there were double-blind tests that were done that gave us this rough range. So again, very interesting, in particular that you're going to have that pelvis. So you have a lot of the fossil looks very much, a lot of Homo naledi looks very similar to Homo erectus. And then you've got that very primitive pelvis combined with the fact that it dates to 300,000 years is, I think, quite interesting. Okay, the newest find that I just published on in the end of last year is site 110, and doesn't have a fancy name that we put out yet. So here is where, once again, a map. So here's site 101 is here. And so this is kind of behind it. There's an area known as the chaos chamber, but 110 is back here. And we have 110, 111, we're up to 115 right now in terms of fossil localities. As we continue to explore the cave, we found other regions. And so from this site, we have 6 hominid teeth and 28 cranial fragments. And they're all from a juvenile individual, a single juvenile individual. We have 2 deciduous teeth, right and left molars, and then we have 4 of permanent teeth that are just coming in to occlusion. So they are unworn, but they would be of the same age of the individual. So from the pieces that we have, we were able to make a rough hypothesis of what this individual would look like, and we dubbed it Leti, is the name of this species. And so I really like, you can see these are the permanent teeth that are coming in. We've just got the deciduous teeth in there. So here's a comparison to the tongue child and then robustus. And as you can see, or maybe not see quite that well in here, but the cranial capacity is very similar to the tongue child, and it even shares some morphologies with the tongue child. Keep in mind, the tongue child is, the Australopithecus africanus is about 2.5 million years old. Homo naledi, roughly 300,000. So again, quite interesting in that regard. The other quite interesting thing I think about site 110 and the fossils that are found there, the teeth is what I focused mostly on, is the consistency that we have across site 110 with the Lesedi Chamber with 102 as well as 101. Very consistent. The teeth are very simple. They're very, again, similar to each other. So all 3 localities suggest very low intraspecific variation, further suggesting that maybe they are a related community that are using this cave to deposit their dead. The other really quite interesting, fascinating part I think about site 110 is that this is the only one where you have a single individual, and we just have the cranial pieces. Now, I will admit that we have only done surface finds in this area. It's a very, very hard to reach cubbyhole, even harder to reach than the Dinaledi Chamber and certainly the Lesedi Chambers. Very, like one of the excavators had to excavate upside down, reaching into the cubbyhole. She was showing some interesting pictures the other day of that. So very hard to reach. I don't know if we're going to excavate in that or when we'll actually get back to do that, largely due to safety. Regardless, it's either that they took a single individual skull and placed it on top in this really hard to reach cubbyhole. And somebody made that effort, or somebody showed up and they dug a hole. They put the body of a roughly 4 to 6 year old individual with just the head sticking out the top. Riddle me that. That behavior is so fascinating. We're learning so much about the behavior of Homo naledi. But again, so the Dinaledi Chamber, 15 individual, Lesedi Chamber, they're somewhat separated, but we have 3, and in site 110, just a single juvenile individual that was found, early juvenile individual is found in there, the skull. Quite interesting. Okay, the last couple minutes, I thought I would talk about some of the implications of the find of Homo naledi. And so one of the biggest implications is that we need to reassess what it means to be in the genus Homo. Some of the characteristics that we use to define the genus Homo is the pelvis, right? And so we have, it's a very modern-like characteristic, right? To having this bowl shape, to not having that very flared compressed front to back or anterior posterior pelvis of the Australopithecine, something that's more derived, more modern-like, and that's what we typically use to help us assign to the genus Homo. One of the large exceptions to that would be Australopithecus sediba, with its very derived pelvis looking very much like Homo erectus. So, but that's something we typically use. That's more of an anomaly for Australopithecus sediba. We typically look at this pelvis in order to help us to assign it to the genus Homo. And again, that obligate bipedalism, that is one of the hallmarks of us. We also look at stone tool production, as well as the increase in brain size. So those are things we largely use to reassess, to define what is the genus Homo. So with our hand tool morphology, we're going to look at both the morphology of the fossils as well as the stone tools that we find. So with our brain size, we have, up until very recently, an increase in brain size over time. And that's what all the fossils had suggested, and that's what people continue to talk about, even in my intro classes when I'm talking about how we have this increase in brain size over time, until you get to the two fossils. We've got Homo floresiensis and Homo naledi. Homo floresiensis shape of the skull is not quite as modern as we see with Homo naledi, but it's very modern-like globular skull with a very small brain size. Particularly very small for 300,000 years old. So that is quite interesting. So again, we can no longer use brain size as a way to define the genus Homo. So that's part of the ways we need to reassess words. Okay, what about the stone tools? As I briefly touched on before, the wrist morphology and some of the hand morphology suggest that they could have, had the ability, that they did have the ability to make and use stone tools. We have no evidence of stone tools right now at the Rising Star Cave system, right? So keep in mind, absence of evidence is not evidence of absence, but we have no stone tools. They would have had the morphology for it. Now, let's combine this with the information that we have sites in Southern Africa, across all along the coast, and some other ones that also have stone tools and have no direct hominid association. So we can no longer assume, based on the date of those fossils, that they were made by modern humans, right? So we don't have Homo naledi at any other site yet outside of the Rising Star Cave system. And so we cannot say that they made those stone tools, but we also cannot reject the hypothesis that they might have. Again, they had the morphology, there was the possibility of that. So it's certainly something that we are looking into, but I think also something that we need to kind of adjust our time frame when we're thinking about what is the genus Homo, adjust our mindset on that. So last thing I want to talk about is coexistence. And so now that we have Homo naledi at 300,000 years ago, we know that it overlapped with modern humans. And so we've got Jebel Irhoud found in Morocco that dates to roughly 300,000 years ago. We just moved back the dates or changed the dates for Homo 1. And so those are time frames that, again, overlap with Homo naledi. And this is the first time that we have two Homo species that are going to be or species that's going to overlap in Africa. So we have this in other areas, again, depending on what you're going to call a separate species or not, but other areas of the world. But this is the first time that we have it in Africa at 300,000 years ago. Now, what happened between the two of them is also something up for speculation. Jebel Irhoud is going to be up in Morocco in North Africa versus Homo naledi is very South Africa. How much interaction they had, we don't know. In fact, the fossil record for what Homo is in Southern Africa is very poor prior to Homo naledi. We don't have a whole lot of fossils, and they're very scrappy prior to, again, Homo naledi. So a lot of debate about what Homo looks like in South Africa, certainly early Homo. And now we have one date at 300,000 years. Again, we're still trying to figure out what early Homo would look like, having more fossils there. But outside of Africa, outside of Southern Africa, we have much better representation of Homo. So what everybody wants to know, where does Homo naledi fit in our evolutionary tree? And the short answer is we're not sure. But this is definitely something that has been revised and discussed ad nauseam. And so it shares a lot of similarities with Homo erectus. In fact, when the papers first came out, there was a lot of conversation about, is it actually just a little Homo erectus? right? Which wouldn't be too dissimilar from what we see with Flores, because floresiensis shares some characteristics with Homo erectus as well. We don't have that same island dwarfing that you would have with floresiensis, but we do have, again, maybe there's an area in Southern Africa, this refugium, that would have cut them off a bit. But again, still, we have several hypotheses. There's no way you can neatly fit Homo naledi into Homo erectus. If you want to do that and you support that idea, you have to dramatically change the definition of Homo erectus, because again, that pelvis is so primitive, and having a very modern like pelvis and being a very strict obligate biped is one of the defining characteristics of what Homo erectus is. So it'd be very difficult to put Homo naledi in there, again, with that very primitive pelvis, as well as some of that shoulder morphology. Some of the more primitive characteristics that we find with Homo naledi just don't fit in neatly with Homo erectus, not to mention the fact that the brain size is quite small for Homo erectus. So this is one hypothesis that John Hawkes came up with. He's one of the leaders of the project. And so, but that's the basic part, is that we're still working out exactly how it would have fit into the whole fossil record. I will also tell you that it shares some morphology, some dental morphology with Australopithecus sediba, as well as Australopithecus robustus, interestingly enough. So some other characteristics, some other hominids within Southern Africa, whether that means ancestor-descendant relationship or some geographic similarities, again, is still something that we're delving into. So we have a lot of fossils. A lot of people say, oh, it's, you know, we just need more fossils. Okay, that's our tagline, paleoanthropologists. We just need to find more. And here we had over 1,500 found in Dinaledi Chamber. As we continue to excavate some more of the soil that had come out of there over the next couple of weeks, I think we're up to 1,800 specimens. We're no longer excavating Malapa, but as we went through those bags, you've got all of Lesedi, and then all the other sites in between we're still publishing on, but then site 110, we have loads of fossils. And this has not got us to our answer. We have not obtained, you know, we're still asking a lot of questions. We'll say that. So with that, I will finish up and say thank you to Harry and Dylan and everybody else, Harry's, for the invitation to present at the University of Liverpool Evolutionary Anthropology webinar series, and to the team. Obviously, I could not have done this. I did not do that all myself. Part of the Rising Star research team, the excavators, everybody for facilitating, and then the funding. So, thank you very much.
Amazing. Excellent. That was just about, if not more interesting than I think we all expected. Thank you very much. Very good. So we do have a couple of questions in the chat already. Walter has asked, is there any geological evidence of Earth collapse to explain the zigzag cave structure?
That's a great question. And I am not sure. Not that I know of. And I'm basing that largely on, I mean, I would guess not, based on the readings from what I remember from the paper, as well as the pictures don't seem like there would be that rock collapse. But I would have to check that.
Excellent.
I will say,
Walter,
it's not these others. We don't have anything that would crush the fossils in any way to suggest that there was rockfall in those areas. Actually, in any of the chambers, nothing has fallen on that. So that would, I guess, be more support. Why would it just be in that one section versus others? But perhaps. Sorry.
No. Excellent. Walter again has asked, was there any pollen analysis of the soil adjacent to the bones to suggest any sort of ritual burial? And were any artifacts found even with this very primitive hominid?
So no artifacts have been found. Yes, there was some pollen analysis that had been done, and nothing had come of that. At least in the most recent conversations I've had, I have not heard of anything that come of that. There's lots of hypotheses for why we do that. Some people think that's a very modern characteristic to put flowers. Some people think it's pretty, it's a sign of reverence. Some people think it's to get rid of a smell. But if you're taking a dead body to a very far back part of a cave, I don't think you're much worried about the smell, right? I mean, You don't want to see anything else chewing on it, but you're also not going to be anywhere near it.
Yeah, excellent. And we've got one from Andrew asking, has any sort of DNA study been done, attempted on those fossils, on any of the fossils?
Absolutely attempted, but that's still being worked on. So the fossils are actually paramineralized. They're not fully fossilized, which makes them a bit more fragile, but also we're very hopeful that we might be able to get some DNA evidence out of it. So that's definitely being worked on.
Excellent.
And the proteomics, I'll say within there.
Yeah, proteomics is becoming more and more important, I think.
Yeah.
I do have a question myself. It is a little bit vague, but concerning the placement of NALEDI in the bush of human evolution. Do you think there is any sort of weight to be given to the idea that we could be looking at sort of breeding populations rather than defined species? Do you think there's any utility in thinking that way?
Absolutely. Yeah, that's a very much a hypothesis that was rooted years ago, actually, in baboons. And so you have these populations, even different genera of East and South Africa, that will come together and interbreed and produce fertile offspring. So they're not, strict species per the biological species concept. And yeah, maybe there was a lot more interbreeding, introgression that would have occurred between species, specimens, I guess, if you want to say it like that, across Africa. We don't have a lot of data right now. I would like to see more fossils. I would like to see Homo naledi at a site outside of the rising star locality. To me, it's fascinating that we have that many that are only found there. And so that is something that we're targeting at some of the other sites that of similar age or in that overlap in that time frame, to try to find where else they might have been deposited. And I think some of the morphology might be able to help us answer the question that you just asked. But I think it's absolutely a possibility and becoming more and more plausible as we find these specimens. Certainly as we get closer to us in time, and the middle Pleistocene, there's so much overlap between the morphology of specimens across the world. I mean, that's part of the basis of the multi-regionalism hypothesis, that you have this overlap. So, and it's too difficult to actually draw those lines. So I definitely think it is plausible.
Excellent, excellent. We've got one from Tim asking, what was the sex ratio in Site 101?
I don't know the exact numbers off the top of my head. I do know they exist. And we look at, because we know that basically sexual dimorphism, that it was very similar to modern humans, roughly 20% between the males and females. And so it's out there, and we can look at some of the teeth. We're looking at largely at size to differentiate between the males and females, but obviously there'll be some overlap. So maybe there isn't a strict number in those papers, but there are some that are quite big and swim, like Dinaledi Hominid 1 is a large, robust male. Lesedi is a large, robust adult male. But between the overlaps that you might have with some of the males and females, I think there is. I don't have a strict answer for it. We know there's males and females.
Excellent. Got another one from Matt Grove. Are there any indications, e.g., from stratigraphy, about the period over which the remains were deposited? Can you say, for example, that there were multiple depositions over an extended period, or that most of the remains were deposited during a single event?
Yes, I neglected to mention that. They were deposited over a period of time frame. So it wasn't just a single event. And that is certainly supported by the multiple areas that we have. But strictly in Dinaledi, you can tell that they wasn't at one period of time. So they went back there multiple times to bury the dead, to put the dead back there.
Excellent. Very, very intriguing indeed. Well, that, I think, are there any more questions that anyone would like to swing our way? No? Excellent. Well, that was very interesting. Thank you very much for the talk, Juliet. That was thought-provoking indeed. And this will be recorded, guys. So if anyone would like to check out the video, it will be on YouTube probably in the next day or so. And if you join us next week, we will have a talk on worked bone assemblage, leather and fur from the Contrebandiers cave in Morocco. So join us for that as well. And yes, excellent. Thank you very much. Thank you.
Cheers.
See you later, everyone.